So last semester, we looked at general embryology from day 1 up to birth, and also looked at the weeks from week 1 up to week 38. And you remember that I said that in the third week, a process known as gastrulation takes place, where the bilaminar germ disk is converted into a trilaminar germ disk with 3 layers, 3 germ layers, and the formation of the notochord. And the notochord remains in the midline of the trilaminar germ disk. So the 3 layers in the third week are ectoderm, mesoderm, and endoderm. And I say that after the 3 layers have been formed, different organs begin developing from the 3 germ layers. And the germ layer of interest today, where the nervous system develops from, is the ectoderm germ layer. Of course, the ectoderm germ layer forms other parts of the body, but one of them is the nervous system. And when we look at the ectoderm germ layer, it is the midline portion of the ectoderm. I repeat, it is the midline portion of the ectoderm which converts into the nervous system. The midline portion of the ectoderm is the portion which is overlying the notochord, because the notochord is in the midline axis of the germ layers. It is in the midline axis of the germ layers. So that part of ectoderm which is overlying the notochord, that is the part of ectoderm in the midline, is the one which converts into the nervous system. So the underlying ectoderm induces the midline portion of the underlying notochord. I repeat, the underlying notochord induces the overlying portion of the ectoderm, which is the midline portion of the ectoderm. It induces it to become thick, and that thickened part of the midline aspect of the ectoderm is what we call the neural plate, and it is the one which is going to convert into the nervous system. So I said that the nervous system develops from ectoderm germ layer. The midline portion of ectoderm, which is overlying the notochord, is induced by the notochord to thicken to form a neural plate. And that neural plate is the one which is going to fold and form a tube known as a neural tube, which neural tube will convert into the nervous system. Those are the introductory points. But let us follow. As I said that this lecture is being recorded. So I've talked about the third week. And I've talked about ectoderm germ layer, the midline of which thickens under the influence of the induction of the notochord and becomes a neural plate. Illustration, let's say this is the entire ectoderm germ layer. We have pulled it out. The enlarged part of the germ layers is the head region, and the narrower part is the caudal region. So you are seeing that this midline portion of the ectoderm is becoming thick. The midline portion of the ectoderm is becoming thick, and this thickened midline portion of the ectoderm will form what we call the neural plate, from which the neural tube or the nervous system is going to develop. These peripheral thin areas of the ectoderm are the ones which are going to form the other parts which are derivatives of the ectoderm. But our focus is on this neural plate, the thickened midline portion of the ectoderm. Now let us make a cross section through the ectoderm and we see how it looks like a cross section. So here we are having ectoderm, this is mesoderm, and this is endoderm. And we are looking at the trilaminar germ disc in cross section. And you're seeing the notochord in the midline. It is the one which induces the overlying ectoderm. And you see this is the midline portion of the ectoderm to thicken and form what we call a neural plate, which we are seeing here in the middle. These lateral aspects are thin and the midline aspect is the one which is thick. This is what we are seeing here. The lateral aspects are thin and this midline portion is the one which is going to become thick and it's known as a neural plate. So it is this underlying notochord which induces this overlying midline portion of ectoderm to become thick and form what we call a neural plate. Now, I want to make another point. The cells on the lateral aspect of the neural plate, where we have painted black, these cells are known as neural crest cells. If I can estimate it in this illustration, the cells in the lateral aspect around there are known as neural crest cells, and we are going to look to look at the importance of these neural crest cells, which are on the periphery of the neural plate. So after our neural plate has been formed, now one may ask, what causes it to become thickened? It is mitosis, cell multiplication or proliferation of these midline cells. So what happens after the neural plate has been formed? The sides of the neural plate where you have the neural crest cells begin to elevate. Now you're seeing them elevating. They begin to elevate. And these elevated parts where we have neural crest cells are the ones we call neural folds. Neural folds. Let's see it. You can see these lateral aspects begin to elevate, and these are what we call neural folds. And you're seeing that the elevation is beginning in the midline. Then you can also see these neural folds. So what happens? The neural folds move and fuse with each other. They move and fuse with each other. And this is what I am talking about. Remember, neural plate, neural folds, then the neural folds fuse with each other. And the fusion begins from the middle of the neural plate. They fuse with each other. Now, before the neural folds fuse, and I want to make this point, before the neural folds fuse, these neural crest cells will separate from them like that. The neural crest cells will separate from them, then the rest of the folds will fuse. And I said that fusion begins in the midline and then approaches into the head region and the tail region like this. Fusion began in the midline aspect, middle aspect of the neural plate, and then it proceeded to the cranially and caudally. And I said also made a point that before the neural folds fuse, the neural crest cells separate. So then the neural folds will finally fuse, and when they fuse, the neural plate is converted into a tube known as a neural tube. And these are the neural crest cells which separated from the neural folds, and they will be converted into the neurons of the peripheral nervous system, the melanocytes of skin, and the medulla of the adrenal gland. Major parts of the peripheral nervous system that neural crest cells will form, they will form the neurons, both motor and sensory neurons of the peripheral nervous system. Then they will also form the medulla of the adrenal gland, and some will convert into the melanocytes. Then the rest of the tube will join, the rest of the folds will join to form a neural tube. And that is what we shall have at the end. After the folds have fused and the neural crest cells have separated, you're going to have a tube known as the neural tube. Now this tube, as the name suggests, has a cavity within and has an opening in the cranial or head region known as the cranial pore, and also has an opening in the caudal region known as the caudal pore. And inside it there is a lumen, and then the cells of ectoderm for the neural folds are the ones forming its wall. And then this wall is minus the cells of the neural crest which separated. Hope that is very, very clear. Remember, the peripheral aspects of the ectoderm are going to form other parts which are derived from ectoderm. So what are we having? Let me show you an example of what we are having. So at the moment, we are having a tube which has formed. However, this tube, as I said, it has a cranial opening and a caudal opening. The cranial opening is known as the cranial pore, and the caudal opening is known as the caudal pore. Those are the beginning stages of the formation of the neural tube. And formation of the neural tube is known as neurulation. Neurulation is the process through which the ectoderm forms a neural tube as a result of the inducting influence of the underlying notochord. So now we are having our neural tube with a cranial pore and a caudal pore. And these other illustrations are also showing us that as the neural tube forms. So you can see this is ectoderm. The neural plate is in the middle with neural crest cells, notochord. Then the lateral aspects elevate to form neural folds. But before the neural folds fuse, the neural crest cells separate, and then the folds fuse to form the neural tube. It has a wall and a lumen and two neural pores, the caudal and cranial. And this is our notochord, which induced its formation, the process known as neurulation. What happens? On day number 25, the cranial pore closes. Let's see. On day number 25, the cranial pore closes. This is very important, and I will show you why it is very important as we proceed. The cranial pore closes on day number 25. And 2 days later, on approximately day number 27, also the caudal pore closes. Cranial power closes on day number 25. Caudal power closes on day number 27. So by day number 27, you have a closed tube. And this is what I have been talking about. Closure of this, day number 25. Closure of this, day number 27, such that now you have a completely fused tube. After that, what happens? Remember, this tube has a cranial part and it has a caudal part. Now, in the cranial part, due to increased growth, 3 dilations form. In the cranial part, 3 dilations form. What do I mean? Let's see what I mean by that. This is a tube, it continues to grow. In the cranial part, you can see 3 dilations form. This, that, and that, due to increased growth. But the rest remains as a tube. So these dilations are located in the cranial part of the tube. And these dilations are forebrain vesicle. They are known as vesicles. Forebrain vesicle, the dilations are known as vesicles, remember. So I have a forebrain vesicle. The other name for forebrain vesicle is prosencephalon. Then you have the midbrain vesicle, which is known as the mesencephalon. And then hindbrain vesicle, which is known as the rhombencephalon. Now, this encephalon means head region of the tube. Pro means superior or first. Mese means middle. Rhombe means behind, hind. Forward, middle, hind. So these are the ones I have talked about in this illustration, in this slide, that relations develop in the head part of the cephalic part of the tube known as prosencephalon, mesencephalon, rhombencephalon. All this due to increased cell division. And the rest of the tube will remain. It will increase in length, but it will remain as a tube without getting dilated. So this tube principally is bent because the embryo is also bent. It's not a straight tube, it's a bent tube because of the bending of the embryo. And you can see the 3 vesicles in the head region, and the rest of the tube remains tubular. It increases in size but remains tubular without getting dilated. Hope you are following. Remember, I am recording this lecture. Let's continue. And here I said at about 5 weeks. Remember, we began the neurulation period in week 3. At about 5 weeks, the forebrain vesicle known as the prosencephalon divides into 2. It was 1 vesicle. Now it is differentiated into 2 more. And those 2 are what we call a telencephalon. Telencephalon is going to have a left and right half, and then the diencephalon. Let's look at it and we see. See? This tube, which was the first dilation, the prosencephalon, now was differentiated into 2. The telencephalon, which has a right and a left part, and then the diencephalon, which also has a right and left part. So originally you have 1, and by 5 weeks it has differentiated into these 2. Then the hindbrain vesicle also divides into 2. It was originally 1, Now it's divided into two. This big one and this narrow one. The narrow one is known as the mesencephalon, and this big one is known as the metencephalon. What about the midbrain vesicle? This one does not differentiate into two, but it just increases in size. So this is what I've been talking about in this slide, about 5 weeks. The prosencephalon divides into the telencephalon with two portions, the right and the left, and then also the diencephalon. Then the rhombencephalon also divides into two. What are they? The metencephalon and the mesencephalon. What about the mesencephalon? It doesn't differentiate into two, but just increases in size. Now, what is going to happen? What is happening here? Their structures I've talked about. Furrows. These are depressions between the dilations. The rhombencephalic furrow or the rhombencephalic isthmus is a depression separating the mesencephalon from the rhombencephalon. Where is it? This one. It separates the rhombencephalon from the mesencephalon. This is the rhombencephalic furrow or isthmus. This is the furrow, and the narrowing here is the isthmus. And that's what I've been talking about. And then there's what we call the pontine flexure. The pontine flexure is that bending between these two derivatives of the rhombencephalon, the metencephalon and mesencephalon. Let's look at them. This one is the pontine flexure. This one. The pontine flexure is between the metencephalon and the mesencephalon around there. And the rhombencephalic furrow or isthmus is this one between this rhombencephalon and the mesencephalon. So let those not disorganize or confuse you. So we moved from here. Prosencephalon has divided into these two, the tele and the die. So you can see this has now increased. It's this one. It has differentiated into these two. And each one has a right and left side. The mesencephalon has just increased in size. The rhombencephalon has divided into the metencephalon and the mesencephalon, separated by this pontine flexure, and the rest remains as a tube. So this is what we've been talking about. Remember, the tube had a cavity. Now, as these vesicles form, even the cavities changes shape. Here the cavity was regular. Now here it has changed shape. Even here it is continuing to change shape. Remember, the cells forming the wall of this tube are derived from ectoderm, and they are going to form the cells of the central nervous system. So it is a tube with a wall made up of ectoderm cells, which are going to form the cells of the nervous system, central nervous system. And these spaces which are within the tube are the ones which are going to form our ventricular system, ventricles and the collecting channels. These are going to become ventricles. That is why the brain has cavities in its core. The brain has cavities in its core, and those cavities are derived from the lumen of the tube, and these cavities are the ones which change shape with the continued growth of the tube, and these cavities are going to form the ventricles and the channels connecting the ventricles, which constitute the ventricular system. So, in the adult brain, what will the telencephalon form? What will the telencephalon form? In the adult brain, the telencephalon is going to form what we call the cerebrum. And this cerebrum is going to be one of our lectures. That folded part of the brain is what we call the cerebrum, and it is derived from the telencephalon. The cerebrum has an outer layer known as a cortex and an inner layer which is white matter known as the cerebromedulla. So the cerebrum has an outer layer which is dark and which is made up darker because it is gray matter known as the cortex, and an inner layer which is white known as the medulla. And because the telencephalon has two sides, one on the left, one on the right, these sides are going to form what we call the hemispheres of the cerebrum, a right hemisphere and a left hemisphere of the cerebrum, made up of the outer cortex and an inner medulla, which is white matter. That is why when we're looking at the cerebrum, we're going to say the cerebrum has two hemispheres, a right hemisphere and a left hemisphere. Why? Because the telencephalon is segregated into two portions, one on the right, one on the left, projecting the two hemispheres. And it will be having an outer layer, which is the cortex, and an inner layer made up of white matter known as the medulla. It will also form what we call the basal nuclei. These basal nuclei, we call them basal ganglia. What are basal nuclei? Within this white matter, which is forming the cerebromedulla, within this white matter, you're going to find areas of gray matter. The cortex is gray matter, it is the outer layer. But even within this white matter, you're going to find areas of gray matter. And those areas of gray matter are the ones we call the basal ganglia or basal nuclei. So whenever I talk about basal nuclei, those are the regions of gray matter embedded within the white matter of the cortex. And we shall have a lecture on this basal nuclei and what their functions are. So the telencephalon will form the cerebrum with its cortex and medulla and its two hemispheres, and these areas of gray matter within the white matter of the cerebrum known as the basal nuclei. These are the parts of the adult brain. And we are going to look at these parts. These are the parts of the adult brain. In your brain, you have the cerebrum made up of two hemispheres with an outer cortex, which is gray matter, and an inner medulla, which is white matter. And within that white matter, you have areas of gray matter known as the basal nuclei or basal ganglia. These are the ones derived from this telencephalon, which is a vesicle of the forebrain. What about the cavities? What will they form? The cavities on the right and on the left are going to be known as the lateral ventricles. That is why you find that each cerebral hemisphere has a cavity known as a lateral ventricle. You have a lateral ventricle on the right and a lateral ventricle on the left. A lateral ventricle on the right and a lateral ventricle on the left. So this telencephalon will form these parts of the brain and this part of the ventricular system. Then what about this diencephalon, which is also derived from the prosencephalon? This diencephalon is going to form the thalamus. We are going to look at the thalamus, what the thalamus is. It is also going to form the hypothalamus. And also going to form the epithalamus. We shall look at the parts of the brain and we shall see where these parts are located. So when we look at the brain and I show this is the thalamus, this is the hypothalamus, this is the epithalamus, remember they derive from the diencephalon, which is a vesicle of the forebrain. And the diencephalon also form the retina of the eye. Remember I said the retina derived from the ectoderm? Yes. What about this cavity of the diencephalon? That is the one we are going to call the third ventricle. Why is it that we do not have a second ventricle? Because already we have two there. The atrial ventricles are two, so the next one must be the third. Remember when we were in class and two people tied on the first position, the next person will be the third because already you have two. So this is the third ventricle, the cavity of the diencephalon. That is why, and I want you to remember when that time comes, the third ventricle is between the thalamus on the right and the thalamus in the left hemisphere. The thalamus on the right and the thalamus on the left, because the thalamus is the derivative of the diencephalon. So these cells are going to form the thalamus on the right, and these ones are going to form the thalamus on the left, and the cavity between the thalami is going to be the cavity of the diencephalon from which the thalamus was derived, and that is going to be our third ventricle. What about the mesencephalon? The mesencephalon will maintain its name, and in the adult brain, we're still going to call it the midbrain. So when I talk about the midbrain in an adult brain, I will show you that now this is what they call the midbrain. Remember, it was derived from the mesencephalic vesicle, which is our midbrain. And the cavity of the midbrain is going to form a narrow channel known as the cerebral aqueduct. And I will show you when we're talking about the ventricular system that cerebrospinal fluid from the third ventricle passes through the cerebral aqueduct. And when we look at the midbrain, you will clearly see this channel known as the cerebral aqueduct, because the cavity of the midbrain from the neural tube is the one which is going to convert into the cerebral aqueduct. Then the metencephalon, which is the first vesicle of the hindbrain, is going to form the pons and the cerebellum. We are going to have a lecture on the pons and a lecture on the cerebellum. And as we are having these lectures, remember that these two were derived from the metencephalon, which is the vesicle of the hindbrain. What about the cavity? The cavity of the metencephalon, which has converted into the pons and the cerebellum, plus the cavity of the mesencephalon. The mesencephalon is going to form the medulla oblongata. We are going to have a lecture on medulla oblongata. The medulla oblongata is derived from mesencephalon. The cerebellum and pons derived from the metencephalon. We shall have lectures on these parts and we shall see where they are located. And we shall remember that they came from these two vesicles. The cavity of both the metencephalon and mesencephalon, this cavity is what we call the fourth ventricle. And we are going to see that between the pons and the cerebellum, you're going to have the fourth ventricle, because that is the region of the hindbrain, and the cavity of the hindbrain is the fourth ventricle. Then the rest of the tube, which did not form vesicles, is going to be our spinal cord. That is why I told you in the introduction that the central nervous system is made up of the brain. The brain has these parts, and the rest, which remains as a tube, is going to form our spinal cord. And the cavity of the spinal cord is going to be the central canal of the spinal cord. So that is how the nervous system develops, from the neural plate, neural folds, neural crest cells separate to form the peripheral nervous system. The folds fuse to form the neural tube. The fusion begins from the middle, progresses to the cephalic region and the caudal region. Then the cranial pore closes on day number 25. The caudal neural pore closes on day number 27. Then the vesicles develop in the cranial part of the tube, forward, mid, and hind brain, which further differentiate the forward brain vesicle into two, the hind brain vesicle into two. The midbrain vesicle remains one but increases in size. And these are the parts they form in the adult brain or adult central nervous system, and the cavities form these ventricles, and which contain cerebral spinal fluid. Now, this remainder of the tube, which forms the spinal cord, and we shall have a lecture on spinal cord. The spinal cord has sensory neurons which are ascending to the brain from the body part to take sensations, and descending neurons which are motor neurons which are bringing from the brain to bring motor impulses to the body muscles. So the spinal cord contains sensory neurons which are known as ascending tracts and motor neurons which are known as descending tracts. We shall look at them in a lecture. So we are going to focus on the spinal cord, and we also see much as the remainder of the tube is the spinal cord, but how does it differentiate? How does it differentiate to form the different portions of the spinal cord? So we are going to make a cross section through the spinal cord and see what happens as it is developing, and is also of importance. So here we have seen our neurotube forming. This is the ectoderm, and we see our neurotube forming after the inducting influence of the notochord. Of course, this is ectoderm, this is mesoderm, this is endoderm, but we have seen our neurotube separating. And these are the cells which form the wall of the neural tube. Hope you are following. They are derived from ectoderm. So when you make a cross section of the spinal cord here, you're going to see the cells which form the wall and a central channel known as the central canal of the spinal cord. So we are making a cross section here, an opening between the central canal and the wall surrounding it. And this is what we are seeing here. You're seeing an opening, which is the central canal, and the surrounding wall. The surrounding wall. So we have made a cross section of the developing spinal cord. Now, originally it is circular, but due to differentiation, it becomes narrowed in the middle. So you can see the wall of the spinal cord and the central canal. These are the cells of the peripheral nervous system which are attached to the spinal cord. And you remember these cells developed from the neural crest. So this is the spinal cord wall. These are the cells of the neural tube. And then this is the central canal, but it's changing shape. Now, what happens? This wall is going to become thicker. Why does this wall become thicker? This wall becomes thicker because the cells of the neural epithelium or the wall of the neural tube, these cells divide by mitosis, so that the wall of the spinal cord becomes thicker. You look at this. These are the original cells. These are the original cells of the wall. These ones. These ones. They are the original cells. These ones. But they divided by mitosis to form primitive nerve cells known as neuroblasts, and then they are the ones occupying this thickness. So the wall of the tube has become thicker because the original neuroepithelial cells of the neural tube have divided to add more cells or more primitive nerve cells known as neuroblasts. So here we are seeing the wall of the tube dividing to form more cells, both on the dorsal and ventral aspect. And these cells here, which are derived from the wall of the tube, are the ones which make the wall of the spinal cord thicker, and that is how it becomes like this thickness. So let's see how these cells differentiate. Now, before I go there, this is a real neural tube photographed using an electron microscope. You can see the folded tube because the embryo is folding as it continues to grow. Forebrain vesicle, midbrain vesicle, hindbrain vesicle. This is the cavity which is going to convert into the central canal of the spinal cord and the ventricles. And you can see these ectoderm cells forming the wall. And then you see the forebrain has divided into the telencephalon and diencephalon. The midbrain vesicle has increased, and the hindbrain vesicle is divided into metencephalon and myelencephalon, and the rest is the spinal cord, which we are going to look at, having a central canal and a wall. And I say these are the cells which divide to form the thickness of the wall of the spinal cord. So the notes are the ones I'm talking about. So what are we seeing here? We are seeing a cross section of the tube. And the tube is the one which I said is going to convert into the spinal cord. Now, I want you to imagine that originally we only have these cells forming the wall of the tube, and the cavity of the tube is the central canal. These other cells are not available, are not present originally. These are not present originally. These ones in the region we have painted blue and red, they are not present. Originally, we only have these walls of ectoderm forming the wall of the tube and the central canal. And these original cells are the ones we are calling the neuroepithelium, which I've indicated here, the neuroepithelial layer. So, this is the anterior aspect, this is the posterior aspect of the tube. The posterior aspect is known as the dorsal aspect, and the anterior aspect is known as the ventral aspect. So what happens? These neuroepithelial cells, which are the original cells which are in existence, they divide by mitosis and form primitive nerve cells known as neuroblasts. Some neuroblasts accumulate in the ventral aspect, and some neuroblasts accumulate in the dorsal aspect. So you're going to find that the thickness of the wall is going to increase because of these new cells which are derived from the neuroepithelium. So this layer which is added containing both cells in the dorsal and the ventral aspect, this entire layer of these cells surrounding the neuroepithelium is what we call the mantle layer. Now, don't be confused by the labels. Mantle layer does not mean only this. No, mantle layer means that all the surrounding neuroblasts which have been derived from the neuroepithelium, this layer is what we call the mantle layer. So the neuroepithelium multiplies and gives rise to primitive nerve cells known as neuroblasts, which are going to increase the thickness of the neural tube in the region of the spinal cord, forming a layer known as the mantle layer. The central canal, which was circular, now has become diamond shaped, with these depressions known as the sulcus limitans. These depressions are what we call the sulcus limitans. So this mantle layer which is formed increases the thickness of the wall of the spinal cord. After the mantle layer has formed, the cells in the dorsal aspect, these ones which we have colored blue, the cells in the dorsal aspect of the mantle layer differentiate into sensory neurons, and the cells in the ventral aspect of the mantle layer differentiate into motor neurons. The mantle layer forms, after which the cells in the dorsal region of the mantle layer differentiate into sensory regions, and the cells in the ventral aspect differentiate into motor neurons. This region of the mantle layer containing sensory neurons is the one they call the ala plate, and this region containing motor neurons, which is ventral, is what we call the basal plate. So you can also say that the mantle layer divides into the ala plate, whose cells differentiate into sensory neurons, and then the basal, the ventral aspect differentiates into, the neuroblasts in the ventral aspect differentiate into motor neurons, which are located in that region of the mantle layer known as the basal plate. However, the roof of the tube is what we call the roof plate, and the floor of the tube is what we call the floor plate. Now remember, there is no mantle layer between the neuroepithelium and the outer margins of the tube. There is no mantle layer there, and there is no mantle layer there. The mantle layers are just on the side. So, with further development, the ala plate containing sensory neurons is going to convert into the posterior horn, and then the basal plate of the mantle layer containing motor neurons is going to form what they call the anterior horn. So when you look at the spinal cord, it has in the middle a central canal surrounded by the neuroepithelial layer, which are our original cells, having an extension on the dorsal aspect known as the dorsal horn or posterior horn containing sensory neurons, and a ventral horn or anterior horn containing motor neurons. Let's look at it. I'm going to show you this. Now, this is our spinal cord. And in the middle, you have the central canal surrounded by the neuroepithelial cells. Then it has a posterior horn, which came from the ala plate of the mantle layer containing sensory neurons. And then an anterior horn, which came from the basal plate of the mantle layer containing motor neurons. When you make a transverse section of the spinal cord, this is what you see. The mantle layer containing the sensory neurons, motor neurons, ala plate, basal plate originally, and then the central canal surrounded by the neuroepithelia. Let's go back. Now, look at this illustration again. There is an outer margin which is colored white. It is not part of the mantle layer. It is on the sides of the mantle layer. This is the mantle layer, but what about this region? This outer region is what we call the marginal layer. Is what we call the marginal layer. The marginal layer is white matter. This is what we call the marginal layer. Let's go back. This is what we call the marginal layer. What is contained within the marginal layer? You remember when we are looking at nerve tissue, a neuron has a cell body, and the cell body is connected to the axon. And what is surrounding the axon? A myelin sheath. And that myelin sheath is coated by a glycoprotein. And the characteristic color of proteins, those of you who did food test, is white. So what is this marginal layer? The cell bodies of the neurons are here in the ala plate and the basal plate. This is where I have the cell bodies of the neurons. But their axons are facing outwards. So this marginal layer, which looks white, contains the axons of this cell body. The cell bodies are in this region. Then their axons are facing outside. That is why this outer layer, because of the myelin sheath or myelinated axons which are in this region, it looks white. So the marginal layer contains the axons of the new cell bodies of the neurons, which are located in the alar plate and the basal plate. So the axons are facing outwards. So that is why this region in the marginal layer is white, because it contains axons with a myelin sheath. What about the roof plate and the basal plate? The roof plate is there and the basal plate is there. And when you look at the spinal cord, you're going to see our roof plate. Let's see where the roof plate is. The roof plate is there and the floor plate is there. So that is how the spinal cord differentiates into the outer white matter containing axons of neurons, the alar plate which forms the posterior horn containing sensory neurons, the basal plate which is the anterior horn containing motor neurons, the central canal surrounded by the neuroepithelium. That is when you make a cross section of the spinal cord. Now remember, we say that it is these neurons which convert these cells of the neural tube forming the neuroepithelium, which were originally present. Then they form these neurons of the alar and basal plate of the mantle layer, increasing the thickness of the wall of the spinal cord. Now I want to make another point. After these cells have formed these neurons, originally they are known as neuroblasts. Then when they mature, I call them neurons in the spinal cord. After these cells have formed the neurons, remember one thing, they are forming the neurons of the spinal cord, because now we are talking about the spinal cord, but in the head region, the brain region, they are also forming the neurons of the brain. What is happening in the spinal cord is also happening in the brain region. So these neurons, after forming the neuroblasts, this neuroepithelium, after forming the neurons of the spinal cord, then they begin differentiating into neuroglia. They begin differentiating into neuroglia. That is how our neuroglial cells are formed. They begin differentiating into neuroglia, meaning that these neuroepithelial cells first form the neuroblasts, which are the sensory and motor neurons of the spinal cord and the brain. Afterwards, they then differentiate into another neuroglia. And remember, during our cells and tissues, we say that the nervous system is made up of neurons and neuroglia or glial cells, and then they begin forming the astrocytes, oligodendrocytes. These are the ones which form the myelin sheath of the nervous system, central nervous system. Then they form the microglia, ependymal cells participating in the formation of CSF, microglia, which are the immune cells of the nervous system. So you're seeing the neuroepithelium is pseudostratified after forming the neuroblasts. After forming the neuroblasts, both sensory and motor, it then divides into the glial cells, ependymal cells, then the astrocytes, then the oligodendrocytes, then some of the oligodendrocytes will have an intermediate which will form the microglia. So this is showing us that it is the neuroepithelium which forms both neurons first, and then the cells of the glia or neuroglial cells. So we have been focusing on how the spinal cord forms and how these different parts of the spinal cord come out, the outer white, then the inner gray matter forming the posterior and anterior horn. Everything begins there in the middle, where we have the neuroepithelium and the central canal of the spinal cord. The same process of forming neurons first and neuroglia second is the same process taking place in the parts of the brain which I talked about: forebrain, midbrain, and hindbrain. So what are we seeing here? Let us focus on this and understand it very clearly. A spinal nerve is called a spinal nerve. Of course, in the neck we have from C1 to C8. In the thorax we have T1 to T12. In the lumbar region of the spinal cord we have L1 to L5. In the sacral region we have S1 to S5. In the coccygeal region we have CO1. They are known as spinal nerves because they are connected to the spinal cord. Number 2, these spinal nerves contain both sensory neurons and motor neurons. Now, when a nerve has both sensory neurons and motor neurons, it is known as a mixed nerve. So, spinal nerves, which we're even looking at in the thorax, T1 to T whatever, abdominal wall, so they are mixed nerves because they have sensory neurons and motor neurons. Motor neurons are going to supply muscle. Sensory neurons are coming from skin and deeper organs, bringing sensations to the central nervous system. So you are finding that this is a sensory neuron. It moves and enters into the spinal nerve. In the spinal nerve, now the spinal nerve, as it approaches the spinal cord, divides into two. As the spinal nerve approaches the spinal cord, it divides into two. A posterior horn, whose neurons are going to go to a posterior root, whose neurons are going to go to the posterior horn. And remember, the posterior horn has sensory neurons. And an anterior root, whose neurons are going to go to the anterior horn. And anterior horn, remember, it has motor neurons. So these sensory neurons enter the spinal nerve. But when they reach at the roots, the sensory neurons enter the posterior root, and through the posterior root, they will connect to the posterior horn of the spinal cord and synapse with ferrosensory neurons. So the posterior root of the spinal nerve contains sensory neurons because they have to pass there on their way to the posterior horn and synapse with ferrosensory neurons. And these ferrosensory neurons will pick the sensations from there and ascend them to the brain for interpretation. Then when the brain sends down motor impulses through motor neurons, the motor neurons in the central nervous system will come down at the level of the spinal cord and synapse with motor neurons of the anterior horn. Remember, the anterior horn has motor neurons. Then the motor neurons in the anterior horn will move out of the spinal cord through the anterior root, and then they will go to supply the muscle. So the motor neurons are connected to the anterior horn and they pass through the anterior root, and sensory neurons are connected to the posterior horn and they pass through the posterior root, because in the posterior root we have sensory and the anterior horn we have the motor. That is the structure of the spinal nerves as they connect to the spinal cord. Now, when you look at the posterior root, which is transmitting sensory neurons towards the posterior horn, it has an enlargement. You see this enlargement? This enlargement of the posterior root is what we call the posterior root ganglion. Remember, a ganglion is an enlargement along a neuron of the peripheral nervous system. Why do you have this enlargement in the posterior root? The sensory neurons are unipolar. A unipolar neuron is one which has a head with one process leaving it, which process divides into a dendrite and an axon. So you find that because in the posterior root, that is where you have the cell bodies. You have very many cell bodies there because the nature of the structure of the sensory neuron dictates that the cell bodies must be around there. So have very many cell bodies accumulating in the posterior root. That is why the posterior root enlarges to form the posterior root ganglion. Whenever you see a ganglion, you know that very many cell bodies are accumulating there. And that is why the structure of the sensory neuron, which is unipolar, the one process leaving the body dividing into the peripheral process. Now, the peripheral process is the one which is connected to skin, and it is acting as the dendrite. And the central process is the one connected to the spinal cord, and it's the one acting as the axon. And the cell body is in the middle in this region of the posterior root. That's why the posterior root is enlarged to form the posterior root ganglion. So you find that by looking at this, the embryology of the spinal cord enables you to understand the structure of the spinal nerves in relation to the spinal cord. Remember, these spinal nerves are part of the peripheral nervous system, and they were derived from the neurocrest cells. Very, very important. And I believe you're now appreciating how the embryology of the nervous system is very important in understanding. So what are we saying here? We are saying sensory neuron picking sensations. Pacinian corpuscles are sensory receptors in skin, and some are in joints. Sensory neurons pick information from the body, take it through the posterior horn. Then when they reach the spinal cord, for example, this is picking sensations from the joints, taking it to the spinal cord. Spinal cord is synapses with another neuron, which takes it up to the brain through the thalamus. And we are going to look at this in one of the lectures. Now, we have looked at how the spinal cord differentiates into the different parts and how it connects to spinal nerves in relevance to its formation. Now, let us look at positional changes of the spinal cord. Positional changes of the spinal cord. Now, I want to make an important point here, that in the third month, that is the first trimester, towards the end of the third trimester. And every point I make is very important, members. It's important to put your heart to it. To put your heart to it. That towards the end of the first trimester, that is in the third month, the spinal cord extends the entire length of the embryo. In other words, the length of the spinal cord goes up to the caudal aspect of the embryo. Let's see it. You see, seeing it is very important. So, you see this is the vertebral column representing the length of the embryo. And this is the spinal cord, and they are equal in length. That is by the end of the first trimester. The length of the embryo and the vertebral column are equal to the length of the spinal cord. I want you to remember this. The different segments of the vertebral column are represented on the spinal cord. With the exception of the cervical region, we have 7 cervical vertebrae, but the cervical region of the spinal cord has 8 segments. That is why you have for vertebrae C1 to C7, but for the spinal cord you have C1 to C8. And I said that C8 vertebra is envisaged to have fused with the skull. That's why you have 7. So the segments of the spinal cord represents the segments of the vertebral column. And by the end of the first trimester, the length of the vertebral column is equal to the length of the spinal cord, in such a way that if this is sacrovertebra 1, exactly opposite to it is segment of the spinal cord S1, because they are equal in length. And the spinal nerve coming from the sacral segment 1 moves out exactly at the same level below S1 because that is where S1 is located. And this is what I've been meaning here. That in the third month, the cord extends the entire length of the embryo, with spinal nerves passing through the intervertebral disc at their level of origin. Where you have intervertebral disc S1 and vertebra S1, that is exactly where you have the sacral segment S1, and the spinal nerve moves out exactly from that point because they are equal in length. Now here I'm saying with increasing age, the vertebral column and the dura mater lengthen rapidly. They lengthen rapidly more than the spinal cord. Now, the growth of the vertebral column, overtakes the growth of the spinal cord, meaning that the vertebral column is going to, and the dura mater are going to grow longer than the spinal cord. Now, I want to make an important point. This does not mean that the segments of the spinal cord representing the segments of the vertebral column are lost. They remain there. But in this case, the vertebral column overtakes the spinal cord in growth such that the vertebral column grows longer than the spinal cord. So I say that hence the terminal end of the spinal cord gradually shifts to a higher level. And at birth, the lowermost end of the spinal cord, which is representing the coccygeal segment of the vertebral column, the lowermost end of the spinal cord, which is the coccygeal segment of the spinal cord, is opposite L3. Why? Because the vertebral column has grown faster than the spinal cord. Let's see. You see that this is the lowermost aspect of the spinal cord, which is the coccygeal segment. Whereas the coccygeal segment of the vertebral column is lower. It's a lower level. Why? Because the vertebral column has grown longer or has exceeded in growth, exceeded the spine. The spinal nerve from the coccygeal segment will have to go down and move out at the relevant level of the vertebral column, which is representing the coccygeal region. Because the spinal cord has not lost its segments representing the vertebral column, it's just that the vertebral column has overtaken the spinal cord in growth. That is why these ones, remember, where is S1? S1 is there. And S1 vertebra is there. So S1 spinal nerve has to come down to the level S1 vertebral column and come out. So you're going to see that the structure of the spinal cord at birth, the lowermost segment is at the level L3, between L3 and L2 at birth. And you're going to see the spinal cord as spinal nerves moving down from it. Why are they moving down? Because they have to move out of the spinal cord at the relevant levels. So this appearance of the spinal cord, it looks like a horse tail. The horse tail has hairs moving from it. And this horse tail appearance of the lower aspect of the spinal cord is what we call the coda equina. Coda equina. So that is what we are saying, that originally they are the same length, but the vertebral column goes on growing, overtaking the spinal cord, such that at birth, the coccygeal segment, or the lowermost segment of the spinal cord, is opposite L3 at birth, and then that is at birth. In adulthood, this lowermost segment of the spinal cord in adulthood will be found at the level L1. What is causing that? That the vertebral column grows faster than the spinal cord. At birth, it's opposite level L3, and in adulthood, it is the opposite level L1. So what do we have below the spinal cord in the vertebral canal? You have the meninges and cerebrospinal fluid. These ends you have the meninges and the cerebrospinal fluid because the spinal cord ends higher. What is the relevance of that knowledge? Whenever they are obtaining cerebrospinal fluid for laboratory investigations, whenever they are obtaining cerebrospinal fluid from laboratory investigations, it has to be tapped below L3. Because if the needle is inserted at the level of L3, it can go into the spinal cord and injure it. So they go down between L4 and L5 because down there you have cerebrospinal fluid without spinal cord, and you only have these spinal nerves. And these spinal nerves are difficult to be injured because they just move out of the way like threads. So the needle cannot easily injure them. So that's why when we are bending your back to obtain CSF during lumbar puncture, the needle they have to count these spinal processes, and then when they reach L4 and L5, they insert the needle there, because at that level there is no spinal cord, and they obtain cerebrospinal fluid. Even when they are doing epidural anesthesia, sometimes they do it during spinal anesthesia. It can be obtaining CSF or injecting anesthesia within the subarachnoid space, or epidural anesthesia. All these procedures are done below L3 to avoid injury to the spinal cord, which at birth is at level L3. Sometimes it can even remain there for a long time. So to be safe, you go below L3 and carry out those procedures like lumbar puncture and the rest, where you're less likely to injure the spinal cord. We end there for today. The next lecture, I'm going to talk about the embryology. The next lecture, I'm going to talk about the congenital abnormalities. We have looked at the embryology today. In the next lecture, I'm going to talk about neural tube defects, or which congenital abnormalities can arise. And these congenital abnormalities can be diagnosed on radiological modality. So the next lecture we shall be neural tube defects, abnormalities which can arise. What can go wrong when the central nervous system is forming. After that, we shall now go to the different parts of the nervous system and look at their gross anatomy or the structures. As I told